Clinical settings often dismiss positive psychology interventions, like gratitude tracking, as superficial feel-good homework, disconnected from pathological thought patterns. This stigma ignores recent randomized trial data showing that structured gratitude practice yields statistically significant reductions in moderate to severe anxiety and depression. Gratitude acts as a targeted neurocognitive wedge, performing a specific clinical function distinct from generic resilience building. It disrupts a transdiagnostic risk factor known as repetitive negative thinking, or RNT, the compulsive cognitive loops that maintain most mood disorders. Moving gratitude from a subjective feeling to a measurable mechanistic protocol allows clinicians to integrate it into evidence-based practice. The enrollment flowchart from the 2019 Heckendorf randomized control trial provides the foundational evidence for this shift in perspective. In this trial of 260 individuals, participants underwent a strict 5-week program consisting of weekly internet modules and daily app-based gratitude tracking. The study was designed to isolate the mechanism of change, determining if the intervention succeeded by building protective resources like resilience or by reducing risk factors like RNT. Researchers utilized the dual pathway hypothesis to analyze which of these two cognitive routes led to the observed drop in symptoms. In modern clinical psychology, establishing the exact mechanism of a cognitive intervention is as critical as proving its overall efficacy. Human negativity bias evolved as a survival mechanism, keeping our ancestors alert to environmental threats. In a modern context, this bias constantly primes the amygdala, often resulting in chronic stress and pathological repetitive negative thinking. Deliberate gratitude practice activates a counter-circuit involving the ventral tegmental area and the nucleus accumbens. This activation triggers a release of dopamine, the neurochemical reward that reinforces the behavior and begins building a positive feedback loop. The prefrontal cortex then engages to regulate emotions and downregulate the amygdala's stress response. Consistent repetition utilizes neuroplasticity to transform this temporary downregulation into a permanent neural pathway. This systematic practice physically rewires the brain's threat detection network. For clinicians, the challenge is translating this neurobiology into a practical protocol that a patient can follow. The GLAD technique provides this structure, requiring patients to daily record one thing they are grateful for, one thing they learned, an accomplishment, and a moment of delight. These frameworks force the brain to actively scan for positive stimuli, effectively weight training the prefrontal regulatory pathways. The Heckendorf style utilized this high-frequency approach, requiring daily digital tracking to supplement weekly clinical sessions. This daily frequency acts as the catalyst for neuroplastic change, forcing the brain to adapt to a constant cognitive demand. Gratitude requires the same rigid, disciplined dosing as pharmacological interventions to achieve structural change in the brain. This chart displays the primary trial outcomes, showing the reduction in repetitive negative thinking compared to the waitlist control group. Immediately post-intervention, researchers recorded a moderate to large effect size of D equals 0.61. Longitudinal tracking revealed an even more significant shift, with the effect size surging to D equals 1.66 at the 6-month follow-up. This drop in RNT was accompanied by significant reductions in clinical depression, generalized anxiety, and insomnia. The fact that effect sizes escalated months after the intervention ended points to a permanent structural cognitive change, rather than a transient placebo. We must look at the statistical mediation models to understand the exact cognitive path that led to these results. This diagram represents the mediation model for depression found in the trial data. The top A1B1 pathway represents the mechanism of reducing repetitive negative thinking. The data shows this path is statistically significant, confirming that reducing RNT is what mediates the drop in depression symptoms. In contrast, the bottom A2B2 resilience pathway, representing the building of emotional resources, was only marginally significant. A similar pattern emerges in the mediation model for anxiety. Again, the intervention's effect is significantly mediated by the reduction of RNT. For anxiety, the resilience-building pathway was completely non-significant. This proves that gratitude heals by subtracting a negative cognitive deficit, RNT, rather than just adding a positive emotional resource. Ultimately, this profound neuroplastic change allows new regulatory circuits to physically override and permanently dim pathological thought loops. The combination of dopamine release and prefrontal regulation breaks the brain's default evolutionary negativity bias. Targeted protocols like GLAD do more than boost a patient's mood. They actively starve the risk factor of repetitive negative thinking. This neurobiological data requires clinicians to change how they prescribe and view positive interventions. Gratitude functions in evidence-based practice as a mechanistic protocol for restructuring the anxious brain.